Hello friends, myself Dr. Sonu Panwar, microbiology faculty. Here I am with the microbiology recall. Obviously, I know that you are not in a mood to listen to the recalls, but still, and obviously, the recall, the memory based in the starting might not be able to get all the options correct. So, it's my humble request to you that you should also see up that what was the correct like option and all. Please don't start up that this was there, that was not there. Just try to tell up that which all options are there in this. Okay, and let's start up with the first question. Okay, and the first question is a one bacterial cell wall image was given up with the pilus and the capsule. Okay, so that was given off, and I suppose this was not the exact picture which was given off, but still you should know that this particular one was having pilus, was having capsule, was having lipooligosaccharides. Okay, it was having lipooligosaccharides. And if you have seen up the video on the Allen Next app, I've already told up that the Neisseria, this particular species, Neisseria gonococci, matlab, and another one is meningitis, that is meningococci and gonococci. Both have these types of things. They have pilus, but in this, the capsule is also being written, and capsulated is mostly the meningococci. Okay, along with those and other things like OMPs, outer membrane proteins, lipooligosaccharides, they all are features of what? This particular meningococci. Okay, so obviously, I suppose they wants to ask up regarding this only in the. Is it fine? So this was there, and if at all something else was there, then you should also tell up that what is there in this. As per me, a bacterial cell wall image was being given off with the pilus and the capsule. And this particular capsule, you know, meningococci is capsulated. Even gonococci is capsulated. And both of them, they are what? They are gram-negative cocci. Fine. They are gram-negative cocci. And these gram-negative cocci, they have these particular structures, which you can remember by the mnemonic CIPLO, which I've told up in the app itself. Okay. So lipooligosaccharides, actually it's a gram-negative bacteria. The lipooligosaccharide will be there. Outer membrane proteins will be there in this. Okay. Along with that, a number of other things like IgA, A1 protease, and all that will be present. So I suppose this was there in the option, and answer to this question is this area. Right? If you want to add upon to this, anybody from the live chat, you can tell me up, sir. That was added in this particular one, and this was not being given off. If anybody wants to tell. Okay. Now, this is the first one. The second one, if we observe, is this, and you can tell me, was it neonatal meningitis or something else being given in the question? Somebody was telling like certain granulomas were seen up, and granuloma infantiseptica. You know, na granuloma infantiseptica. We say granulomatosis infantiseptica. So this particular granulomatosis infantiseptica or neonatal meningitis, if at all is being given off, that is being done by what, guys? That is being done by listeria. Okay. And the students were telling that the options was having this particular thing, that the organism is motile at 20 to 25 degrees centigrade and it is non-motile at 37. Okay. So obviously we are talking about the listeria, and which type of motility is present in this? Answer is tumbling motility, guys. Tumbling motility is observed in listeria, and this is the disease which is there, granulomatosis infantiseptica, in which the nodules are observed up over the spleen and other visceral organs, and even the abscess and all can also be formed. Okay, as the name suggests, there is a formation of granulomas in the infant age group because of sepsis, and it is being done by what? Listeria monocytogenes. Right? And this particular thing, if you like seen up my notes or my app, in that I've discussed up that this particular feature is termed as differential motility. Okay, this is differential motility based on temperature, and this differential motility is tumbling type. And this particular thing is also seen in Yersinia enterocolytica. This is also the next, it will be coming up in this way, that Yersinia enterocolytica is the other organism which shows up differential motility based on temperature. Is it fine? So I suppose this was there. You can tell me, sir, what is actually the disease being given off. But if it is so, then obviously the answer is listeria, and this differential motility is observed in that. Is it fine? Now, somebody was telling that this was asked, a history of a man was given with a renal transplant, and that fellow was on some end, like was having end stage renal disease before, and he or she was on many medications before. His hand was red, okay, and then it have involved up the full limb, okay, that is spread onto the whole hand, and this particular stain was being done, and the fungal organism was asked, okay. So I was not knowing up the options. Some were saying blasto is there, sir. The blasto is a dimorphic fungus. Histo is a dimorphic fungus. Some were saying crypto also. And later on, one guy told that Talaromyces marnifi was also there in the option. So guys, this is for sure. If it was there in the option, the answer is this. And I've already told her that this one is common in HIV positive or immunodeficient individuals. And this produces red color pigment. Okay, this particular one produces what? It produces red color pigment. Fine. So this is Talaromyces marnifi, which is also a dimorphic fungus, and this one is common in HIV positive or immunodeficient individuals, and it is producing a which type of pigment? Red color pigment. Fine. And because of that only, I suppose it was starting up red, and later on it involves the whole hand. But I don't know the exact question. You should tell me up, and then we can correct it off. Okay? Because as you know, this is just a recall. So you should also help us in framing up the options. Okay? A 35-year-old female with history of multiple sexual partners presented with a painless ulcer, and there is a history of previous stillbirth. What is the first investigation that you will do? Or like you can also tell up in the chat box that what was there in this particular question. So this is for sure that painless ulcer, painless genital ulcer, if it was there, and multiple sexual partners, obviously it is going towards the STD. Okay, and there is a history of previous stillbirth. So that means that is a known case of first. I suppose so. If at all they are asking regarding that only, syphilis. What is the first investigation that you would like to do up for the successive pregnancies and all that is like things are improving and all that? The prognosis of the syphilis is based upon a test which is termed as VDRL test, which you know is a slide flocculation test. And I suppose that was there in the option, Mineral Disease Research Lab. Okay, yeah, very good med psycho. It is, I suppose, VDRL only that was there. The culture from the ulcer base, nobody will do a culture from the ulcer base for this. And ELISA HIV does not have any sort of relevance here. You know that principle, Kasovich principle is there, that successive pregnancy, the prognosis of that particular newborn becomes well. So that particular thing applies here, and the test helps us. Same thing is for neurosyphilis, investigation of choice is what? For that also CSF. So that's a extra point which I've told you. Thank you, Ankit. The same question it was, huh? So VDRL is the answer of this one. Okay. Now, coming to this, that the incubation period was being asked. Okay. Coming to this, the incubation period was asked in the question. And important here is, and I suppose it was a match the following. So syphilis, you know, I have already told you 9 to 90 days. And I suppose in the exam they have given of this 90 days and all. Fine. The chicken pox, 21 days. SARS-CoV, it is like variable. It is 3 to 10. And the upper range is 10 days. Hepatitis A is having 15 to 45 or 15 to 50. If it is given off, that is also fine. So all these options, I suppose, was there in the match the following stuff. And all are in this particular way. You should remember of. Okay. So syphilis is 90 days. Chicken pox is 21 days. Actually, all the fever with maculopapular rash, they all are having what? 14 to 16 days. Okay. But the upper range is definitely towards the 21 days. And SARS-CoV, this is 10 days. And hepatitis A, I already told you 15 to 50, 50 to 150. Remember that mnemonic. Okay. So that is 50. Right. So syphilis is 90, chickenpox is 21, SARS-CoV is 10 days, and hepatitis is 50 days. All options are correct. I suppose it was coming as a match the following. Ankit, yes or no? Was it there? Med psycho? Yeah. Okay. Now, this was there. Achha, micro questions were lengthy. Fine. So basically, I could able to get 3 to lines. So basic purpose is to tell you the correct one and to have the correct options also from you. So which of the following is not transmitted perinatally? Okay, this question was there. TMV was in the option, rubella was in the option, herpes simplex virus in the option, hepatitis B was in the option. Okay. So perinatally, if you remember of like rubella in the first trimester or up to 20 weeks, it's very dangerous. After that, it does not cause any of the problems. So I suppose if the question was in this sense, the answer is rubella. Because CMV does cause perinatal transmission, HSV is there, hepatitis B is also there. So all of them is there. So answer to this question, Ankit, as you have written up, rubella, Mohit, it's good that you were able to do PGI level, sir. Okay. Ankit, you are telling PGI level. But I suppose if you go deep up, na, like if you see up the things and correlate with the clinical stuff, then you will able to do up. Let me show you 2, 3 questions which have come. That Penicillium marnifi, it was there, like Talaromyces marnifi. So that particular question is really good and it was a repeat. Previously also it have come in the INI-CET only and I suppose in the NEET PG and all, but it was a repeat one because it is produced reddish pigment common in HIV positive ones and I suppose that particular one wants to ask her. Okay. So to this, I suppose the answer is rubella. If it is this particular question. Priyanka have told rubella is the answer. Yes, you're right, Priyanka. Now see this. This is an interesting question which have come. A critical care specialist, like infectious disease specialist, and nowadays in the ISU, a lot of microbiologists are also doing up this particular work, has grown organism in different culture, observed resistance to the carbapenem, fluoroquinols, aminoglycosides, and is it written that it is susceptible to cotrimoxazole, ticarcillin, and clavulanic acid? Huh? Is it written? Yes. Yes, please. In the chat box, if anybody can tell that is it written in the question that it is being susceptible to the cotrimoxazole, ticarcillin, okay, clavulanic acid. Was it there? Parad, what you are telling triple C is what? You are writing the answer as Acinetobacter baumannii. This particular question seems tricky. Okay, this particular question seems tricky. And I suppose somebody was telling that it is also being written that it's a gram-negative bacteria. Okay, it is also being written of that it's a gram-negative bacilli being written in the question. Susceptible to cotrimoxazole, ticarcillin and clavulanic acid. This particular thing is a strong feature of Thinotrophomonas multifila. It's a controversial question. Okay, gram-negative bacilli was being written off. So, if you have seen the recent lecture of mine, I have told up that this particular bug is coming off Thinotrophomonas multifila and the full name is Thinotrophomonas multifila. This particular organism is intrinsically resistant to number of drugs like these carbapenems, fluoroquinols, aminoglycoside. It is resistant to them intrinsically only and interestingly susceptible to cotrimoxazole, ticarcillin and clavulanic acid. Okay, so this is very clearly written in the micro book that it is susceptible to the cotrimoxazole and ticarcillin clavulanic acid combination is used to treat this particular infection which is common in the ISU nowadays. Much of you must have marked up Acinetobacter baumannii, I suppose. Have you marked up Acinetobacter baumannii? So Acinetobacter baumannii is sounding true, okay? But only thing which differentiates it from the other ones is this. It is not susceptible to cotrimoxazole also. Which one? Acinetobacter baumannii. And if you see up, if you've seen up my videos, I have told up that this is also a very interesting bug which is found to be multi-drug resistant to number of things, but it is also resistant to cotrimoxazole. But what you can use in this is this combination you can use, that's for true. But it is being written, I suppose, susceptible to cotri. That's why the answer will go in this particular. But still the fingers crossed. What is the exact question? I don't know. If this was not being given up, then the answer is Acinetobacter. Okay. Which is, Acha, Priyanka have marked A and Priyanka most likely it is what is the answer. Right? So these types of, this is a very good level question. This is very good level question which we asked up. Now, this is a simple one. I've also told up a woman resident in the gynae OPD with a complaint of vaginal discharge examination. Clue cells were found in the microscopy. What are these clue cells? So I already told up that these are epithelial cells with the organisms over them. Okay, they replace up the normal flora, and the most common is Gardnerella vaginalis. Okay, and this is giving a strong clue regarding the bacterial vaginosis, and that's why we told them what clue cells. And you have that EMSA's criteria. Fine. The pH goes more than 4.5 and all those particular stuff, that there is a grayish white discharge. Along with that, if you add KOH, the whiff test, the fishy smell comes. Okay. And this particular clue cells is a feature. The answer is bacterial vaginosis. And here, I suppose you would have done this easily. Right? Now, there was a question regarding this particular one. A child is there with slap cheek appearance, likely to develop which particular ailment? Is it pure red cell aplasia or Kaposi sarcoma? Other ones you can tell. Med Psycho Priyanka Ankit, can you tell me up what are the other options in this particular one? One is pure red cell aplasia, Kaposi sarcoma. Answer is very firmly pure red cell aplasia. Okay. And slap cheek appearance, everybody knows, is being done by this parvovirus B19. Okay. And this parvovirus B19 is a single stranded DNA virus. It's a single stranded DNA virus, and it is non-enveloped. Fine. So pure red cell aplasia is what? Erythroblast, they could not able to be formed up. Okay. And anemia happens. Patient. And basically, this is like one of the common causes of this is infection. Infection by which virus? Parvovirus. Okay. And this already have told up. Acha, the C option is molluscum contagiosum. Fine. Molluscum contagiosum is one of the options. Okay. Fine. So single stranded DNA, non-enveloped. This is also important, guys. This can come again somewhere. And it is having a slap cheek appearance, erythema infectious. And you know that we have done it. That is, I have told up now that particular mnemonic also apne par aaye. So aplastic crisis, polyarthropathy syndrome, NIHF, okay, erythema infectiosum, erythema infectiosum is slap cheek. So that you already know. Fifth disease. Now this you tell, guys. In toxoplasmosis, which is seen? In toxoplasmosis, which is seen or which is the infective form that is being asked? Okay. This is the infective form, I suppose, which is to be asked in this. Yes. Tell me about this particular question, guys. That in toxoplasmosis, which is the infective form done by Toxoplasma gondii? I'm waiting up that. Can you tell me the options there? What is there in the options in this particular one? Infective form, it was asked. Is it sporozoites being written off? Is it sporozoites written up in one of them in the fecal material? In the cat feces? Ashish is telling bradyzoites. So guys, bradyzoites is not the answer. If it is the cat fecal material infective form for the humans, it is being asked. And is the option oocyst was there? Is the option oocyst was there? Yes, please. Is the option oocyst is there in this question? Priyanka, Ashish, Ankit, can you tell me? Gametocyte was an option. Okay. Gametocyte. Oocyst was not in the options. Locust. The answer to this particular question, guys, as per me is oocyst was there. But if you have seen the lectures, I have told up a simple oocyst is not infective in the cat fecal material. It is the sporulated oocyst which is infective. It is the sporulated oocyst which is infective. And this sporulated oocyst is having what inside? It is having sporozoites, guys. Okay. So, answer to your question will be sporozoites. Ankit, oocyst is there, but that was not the answer. I've already told her that oocyst is not infective. Infective is sporulated oocyst. And what is sporulated in that? The sporozoites. And sporozoites is, I suppose, was there in the option. So I suppose the answer is sporozoites. Infective form it is being asked. So I have already told up in the notes also or in the app also. Sporulated oocyst, tachyzoids, bradyzoids, these are the ones which are infective. Otherwise, the oocyst simply is not infective. And that's why they have asked up. And I have told they will try to dodge you with this. They have done this. Okay. So even the examiner is also seeing up the videos. Now, I suppose this was there, the acute branching with septate hyphae. And was it a clinical scenario what they have given up in this? And then they have asked up, Yatin, Ankit, Jay, the paper was tricky. Vikas, yeah, true. Paper was tricky. So can you tell me, this was their acute branching with septate hyphae? And in what particular language it was there in the question? Acute branching with septate hyphae. Yes, please. Anybody? Jay, Shreya, Priyanka. So what was the answer? J of what? Which question? Last one. Last question. Sporozoites is the answer. Yeah. Achha, it was with image. Okay. This was with image. Question was with image. It was a image based MCQ. So acute branching, I've already told up with the septate hyphae is typical of Aspergillus. You know that. Dichotomous branching, mostly the angle is 45. It can be also acute angled. Okay. And somebody was telling that there was a question regarding mucor also. Achha, Aspergillus clinical scenario was being given of gross of lungs. Okay. 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 Now, regarding mucor, can you tell me, guys? So somebody was telling that mucor was also being asked. And it was asked that A septate hyphae at 90 degree. Yes, if anybody can help up in this. That mucor was also being asked. कोई नहीं जय. लेकिन मैंने ये like I have told this particular thing in the app itself or in the live class also. They will try to dodge you with oocyst. Correlated oocyst is the answer. Acha, mucor was asked. Acha, image based question only. So for mucor also it was a image based. Okay. So was it written A septate hyphae 90 degree? Was it written? Amir told yes. Acha, the options were given phycomycosis. Phycomycosis. Mucor was not there. And the phycomycetes is this only, na? Zygomycetes, phycomycetes. So that will do. Answer is that's one. Phyco. Any other question, guys, in the chat box if you want to tell up which have come in the micro and you think that कोई नहीं जय अब कर लो. I am on Ellen next, so you can have that app, right? Or in the app also we are having like simultaneously the live session. You can check upon other subject videos there in that particular one. Okay, there was no mention of mucor. Fine. So FICO is fine. FICO is fine. And the J answer is FICO. Okay, guys. A very best of luck from my side. And do tell up which other micro questions you have observed up, infectious disease questions which you have observed up, anything related to HIV, hepatitis and all, something if you remember of is there that you can tell up in the comment section. RSV infection. I suppose it was being written in the RSV infection that like clarithromycin and all that was there in the option. As such, in the RSV, it causes acute bronchiolitis. Acha, okay. There was a tube agglutination test. Okay, okay. Tube agglutination test and it was being asked up regarding the prozone phenomena, I suppose, है ना? Prozone phenomena. I think because the cutoff will be less only because paper is balanced and paper is slight bit tough. Achha, hepatitis B curve also came. Okay. So that was a picture based one, Jay. Which of the following is nutrient agar? Nutrient agar photo was also there. Tube agglutination test. Initially it is found to be negative and later on it's positive. And if you see my videos, this prozone phenomena I have already told up. Seen in which particular one? Brucellosis. Okay, this particular prozone phenomena. Yeah, very good, Samir. It is excess of antibodies and that is observed in what? Brucellosis. If that has come, wonderful. Anchor paper tha, sir. Very good. But I suppose you were able to do up this. Palmer's disease was asked. Leptospira was asked. Leptospira was asked. Triple R. Rat, rice, rain. Palmer's disease. Icterohemorrhagic fever. Was there? Was this question there in the exam? Acha. NTHBC. And that particular thing always given. Fine. Okay, what was this Leptospira questions? Now some new big bacteria names were there. Ankit, I've told na that in this particular new one, in the Allen Next app, I have already told up regarding Stenotrophomonas bultophylla. And in my live class also told that this question might come because it is a new bug, and that's why these particular AIMS and the PGI and all, they go towards the research stuff, and this is very common. Okay. The Leptospira was not there, I suppose. And hepatitis B curve, they want to have this IgM NTHBC. They want to know about this IgM NTHBC. He's old. What about the nutrient agar photos? Was there any photo of nutrient agar? And guys, anything is from this. No lepto was there. There was a photo of some bug too. All are bugs only, right? Meghna. Can you tell me regarding something like sterilization disinfection? Was there any question regarding sterilization disinfection, guys? Sterilization disinfection, anything? Anything regarding bacterial genetics and all? Was there something from Journal Microbiology? Yes, anybody? Orantia susugamosi. That this was the given one. Okay. Anything else? Scrub typhus. No nutrient agar. Okay. Okay, Ankit, got that. Anything else, guys? Sputum disinfectant. Can be cresol, it can be phenol. You can disinfect it with the autoclave. That is also there. But immediate one is you can use any sort of this phenol element, disinfect the sputum. Anything else? Anything regarding COVID and all? COVID was asked. SARS coronavirus 2 something. Incubation period we have already discussed up. And guys, for this I have a video on my YouTube channel also. You can see that incubation period video. Again, the question will be coming up because this is favorite of examiner. They keep on asking regarding it. Do download up the Allen Next app also, and simultaneously the session is going on in that particular app itself. Okay. And Sparrow, Jay, Ankit, thank you so much. Like you are being in discussion, you have contributed a lot. And Priyanka also, I suppose. Ed Psycho, thank you so much. And very best of luck from my side. Zyada load lene ki zarurat nahi hai, like which all questions have got wrong. Doesn't matter. You have given your best. And chill today. Bye-bye. Take care.